Hi, thank you everyone for coming and listening. So my name is Rachel Paredes. I'm majoring in neuroscience, and today I'm going to be discussing my current project, which is stimulation of the nucleus tractus solitarius during respiratory allergy. To provide an overview of my presentation today, I'm going to firstly be discussing the neuroimmune system, leading into microglia, which are cells of this system, as well as allergies, the effects of respiratory allergy on microglia, the vagus nerve as a possible route of allergic detection, my research question and hypothesis, and then my experiment design, methods and materials, as well as my progress so far in this project and my expectations. This beginning portion will be a summary of my past work as well as the background for this project, and it will lead into how it influenced my current project today. So to go over the neuroimmune system, it's essentially an overlap between the nervous and immune systems. The nervous system consists of cells that are called neurons and glia, and it has a variety of functions. These cells allow us to interpret sensory information, coordinate bodily movement, monitor organ function, and also responds by electrical and chemical stimulation. The immune system is made up of a number of immune cells. They're very broad, and it functions to detect pathogens and also cellular damage and defends the body from infection. It also surveys the body through circulation. And the combining of these two forms the neuroimmune system, which essentially consists of pathways that allow the two systems to communicate, and also includes immune surveillance in the brain via microglial cells. So these microglial cells are very important, and I'll be discussing their function now. So microglia function as surveyors of the brain. They have a variety of roles. They participate in synapse remodeling as well as neuroinflammation. They help facilitate neurogenesis. And in general, they do this by surveying their microenvironment through the use of these cellular protrusions. As you can see on the image on the slide, it's a tracing of a microglial cell in the hippocampus. And so with those ramifications, they're able to essentially probe their environment and dynamically respond. They exhibit sexual dimorphism. So female microglia exhibit a high response to inflammation, as well as higher tissue repair function, whereas male microglia have been found to have higher antigen presentation ability and microglial density in certain areas of the brain. So with microglia, the lab was interested in seeing whether or not a peripheral adaptive immune response could affect it. So in particular, we were interested in asking whether or not allergies as an adaptive immune response in the periphery could affect microglia. So what are allergies? Well, allergies are a type 1 hypersensitive reaction to allergens. Allergens are what induce an allergic response. And in this allergic response, there are immunoglobulin E antibodies that are released and allow this pathway to continue and allows the body to essentially hyperreact to what's supposed to be non-infectious and not bad. So it causes a recruitment of T cells and also produces inflammation that can become systemic when chronic. What inflammation is, it's a high concentration and migration of immune cells, cytokines, and chemokines. And in specific, a respiratory allergy is just a route of allergen exposure in the nose and respiratory tract. And it's been found before that nociceptor binding to an allergen provides a neuromodulatory effect. So back to the question of what happens to microglia during respiratory allergy. This is essentially a summary of what my previous work was focused on. And so after a morphological analysis of how microglia respond, we found that essentially morphologically there is little to no effect on microglia. So then that leads us to, well, if there isn't any detection of allergy by microglia in the central nervous system, what about the vagal nerve and how it essentially monitors organ function, such as the lungs? Could this possibly be an alternate pathway of neuroimmune communication of allergic inflammation? So in order to go more into what the vagal nerve is, it is a part of cranial nerve number 10. It monitors and coordinates organ function, specifically for breathing and in turn the lungs. And it supplies sensory neuron communication and innervation to the lung. It's organized into ganglia. They're called the jugular and nodose ganglia. And they've been found to fire action potentials following allergen sensitization and exposure. Specifically, nociceptor neurons in the lung express antibody sensing FC receptors for IgE, immunoglobulin E, and also a different type of immunoglobulin. And they have been found to contribute to allergic airway inflammation. Also, importantly, they synapse onto the nucleus of the solitary tract. And so this finding leads me to my current project and my research question, which asks whether or not respiratory allergen exposure differentially stimulates the NTS. My hypothesis is that aerosolized allergen exposure will, in fact, increase neural activity in the NTS. And so my objective, therefore, is to establish whether or not the central nervous system can detect allergic inflammation in the lung. My experimental design consists of a control and an experimental group. The control group consists of mice that are untreated and specifically with filtered air. So the samples for the control group are collected at adulthood, whereas for the experimental group, they are treated with aerosolized fungal Alternaria alternata particulates. This is our allergen in this experiment. And the way we set this up is we enclose them in a chamber with this contaminated Alternaria alternata air for 7 days. And then once again, the samples are in fact collected at adulthood. So I included this little schematic here to provide a better idea of what this chamber looks like. There's Alternaria liquid that is just diffused into the environment. And so this allows the mice to freely move around and not be stressed. In other allergic experiments, usually there's sensitization, which includes injection, but we didn't want to do that here. So this is the way that we've set up our experiment. For the methods and materials, first we had to find the right mouse treatment. As I've already explained, we will be using Alternaria alternata allergens, and the control group will have filtered air. For sectioning, we collected the brains at adulthood, and specifically we're sectioning for the regions of the Botzinger/pre-Botzinger and nucleus tractus solitarius regions. The Botzinger/pre-Botzinger is another big medulla oblongata collection of neurons that participate in respiratory rhythm. So we're also interested in seeing if there's any changes in these areas, specifically for the antibody staining. So I'm going to be staining for CFOS, which is an intermediate early gene and a neural activity biomarker, as well as tyrosine hydroxylase, which is just a dopamine precursor enzyme, but it's specific for the nucleus tractus solitarius. So I'm going to be co-localizing them and seeing how they impact, or seeing how the neurons, their actions during the respiratory allergy. This will be followed by confocal microscopy at 6 degree magnification, and then quantification with Fiji, which is just a software that allows us to quantify the fluorescent antibody staining. So for my progress and expectations, so far I've been sectioning and trying to find the target areas. It takes a little bit of practice, but I have an image here of so far one section that I've collected on the slide, and also the target sort of what the nuclei in the brain looks like when I have to find the target area. And my expectation is that the CFOS expression is going to increase in Alternaria alternata exposed mice, thereby showing that neurons in the nucleus tractus solitarius are stimulated more during respiratory allergy, indicating that the central nervous system is detecting allergic inflammation in the lung. For my acknowledgements, I'd like to acknowledge the Center for Glial Neuronal Interactions. This is what the Carson Lab is a part of, and also the Mark UCR program, as well as the National Institutes of Health for allowing me to fund this very interesting and fun work. And that's my presentation. Thank you so much.
Thank you, Rachel. Again, let's go ahead and keep our questions